a great pleasure to have Ron Vale today for a seminar. And I just want to say that he's not only giving a seminar, but he's also coming to work for us because he's part of our International Scientific Advisory Board, and that will take him a couple of days to pick his brain to help us. So what I should say is he doesn't like his picture. And I understand because he was born in Hollywood, and so you would expect to have a star picture much better than that. And so I'm sorry, we'll get a better thing for the next time. And so he's from California, professor of cellular and molecular pharmacology at the University of California, San Francisco. And his research has focused on molecular motors, and so that has been of interest to a number of people here, like kinesin and dynein. And he was awarded the prestigious Albert Lasker Basic Medical Research Award in 2012, together with Michael Sheets and James Pudish. James Pudish was also a member of our SAB in the past. And so I think there's a long tradition of this area of research to be connected to the Curie Institute. He's also a fellow of the American Academy of Art and Science and a member of the National Academy of Science. He has been an investigator with the Howard Hughes Medical Institute since 1995 and president of the American Society for Cell Biology. So having said that, I should just get back to how he started with a BA in biology and chemistry from the University of California. Then he entered in an MD PhD program at Stanford. And I think this is a very interesting program that I think should inspire us in the way you can train medic and researcher in a manner where in the long term they can either continue doing medicine or do research, but really keep in line with the thinking and the way of looking at things that could be shared between the different communities. So he then graduated in the lab of Eric Schutter, where he pursued his work on how biomolecules are transported into the nerve cells. And that was a time when he visited the Woodhall's laboratory to pursue his studies on a really famous model, the squid giant axon. And then with this model, he was able to observe the movement of organelle along microtubule filament in a manner that is similar to what had been seen by Michael Sheets and James Pudish with myosin. But he also saw that in this system, there was an independence of the movement of the myosin and actin system. So that was really a distinct aspect. So he went on to purify kinesin and then look at the force-generating properties of the system. Then, after his PhD, he returned to the Marine Biological Laboratory for his postdoc. And he joined the UCSF faculty in 1986. He has been using a system that we also love a lot, many of us, the plastic beads to track the motion of motors moving along microtubule and ask force-generating action of kinesin. So this aspect of looking at forces and mechanobiology in a way was something that he has really developed early on. So bringing a concept and thinking from the physics into biology was something very important. He also went to the molecule at the level of the crystal structure with the analyzing motor domain. And since then, he has contributed in many ways to the field of cell biology. And I will not list all of them, but I think it's really exciting that he will discuss his recent work, at least in part, and other things, surprises, here related to RNA aggregation. We did his paper on that in our journal club, so I'm sure you'll have lots of questions. Be prepared, and I can't wait to hear you. Thank you very much, Ron. And I forgot to say, for open science, he's the person to talk to.
Well, thank you. It's great to be here. This is like a second home. I had a very pleasant 1 month at the Curie. My daughter Sophie has worked here. She's probably better well known. Wanted her to be a Parisian for life, by the way. I think she's dying to come back here at some point. And I'm also always look forward to the SAB meeting. So anyway, you have a wonderful place here and a place to do science. And I think all of you are fortunate to be here, whether you're students or faculty. So I want to get away from this picture as fast as possible. This is what happens when, you know, my wife Karen is not looking after me and seeing my hair is combed. But anyway, I just want to put up an advertisement about, well, this is an open science project that I started in 2006, which is really to make scientific talks freely available throughout the world. You know, of course, the Curie has many great seminars. You have a budget to bring in speakers. It's a great, Paris is a great place to visit, so you can attract many people. But, well, first of all, not everyone can come to the Curie. And second of all, you realize that many places around the world that don't have the funds or really the opportunity to attract many scientists. So this project is one in which we film, this is just something more visually interesting than my seminar, but we film speakers in professional studio environment and put them on the web. And so if you haven't seen this site yet, I would highly recommend that you have a look. There's about nearly 500 talks on there. And if you look at the list of speakers, it's a very, very impressive group of individuals. We're trying to also develop this project more in Europe as well, and filming speakers at EMBL. They are partnering with us. So anyway, it is a global project. Actually, most of our views are outside the US. If you want to subscribe to it, all we do is send you, you know, new talks when we release them. But you know, it's just a great way to learn about a lot of different kinds of science as well, on demand. The other open science project I've been involved with is a project called ASAP Bio, which means Accelerating Science and Publication in Biology. And I think I gave a talk on this couple years ago when it was all very new, and I talked about preprints. At that time, hardly anyone knew what a preprint was, I think, when I gave that talk, and it certainly changed quite a lot in the last 2 years. It still has a long way to go, but it's actually starting to, you know, take off in the life sciences. I want to make everyone just aware of another event that we have coming up, which is about peer review. So, you know, this is also a very important topic. It's co-sponsored. We have a meeting coming up with HHMI, the Wellcome Trust, and this is going to be live streamed, so you can follow it in real time in February. But again, you know, I think these are really important issues that are, you know, our scientific community needs to take ownership of. You know, these are things that we just can't outsource to other parties to think about. We have to really be thinking about how we want to communicate our science. And I think this is all part of an effort to really invigorate and energize our scientific community to think through these issues. Okay, so the science. I think I'm going to be a little bit ambitious, which is something that I usually, you know, recommend to my students and postdocs never to do, but, you know, to cover 2 topics in one talk. But the top subject is already published. As you've heard, it's already been done in journal clubs at the Curie. So I also wanted to feature some unpublished work here. This involves more organoids and stem cells and so forth, which is very pertinent to what's going on in this building here. And I know is of great interest in the Curie, so I thought I'd try to talk about both. I want to give these two credit because really I had almost nothing to do with any of those two projects. If you have really exceptionally good people, you know as a PI, the best you can do is just water them gently and then let them grow and do their thing. And when you have postdocs who are as exceptional as these two, gosh, it makes one's life as a PI extremely easy. And I'm not sure who's learning more from one another, but I certainly am learning a lot from both of them, I can say. So I want to just acknowledge that they really drove, in terms of conception and execution, all of the work that I'm going to tell you about. So I'll be a little bit brief on both of them, but I'll try to give you the general idea. So on the first topic, with neurodegeneration, we know very well that there are many cases where protein aggregation appears to be an underlying cause of neurodegenerative disease. And these just show some examples of diseases, and on the right, proteins that have aggregation phenomena that are thought to give rise to those diseases. And you know, there's an enormous literature on all of these subjects. But what I hope to suggest is that in addition to proteins, that RNA also may aggregate, and that this phenomena also could be a contributing factor to neurodegeneration. I mean, this is a very new idea, so you know, it will remain to be seen exactly how it plays out. But I'll give you the story that that developed in our lab with regard to this. So the group of neurodegenerative diseases, actually not just neurodegenerative, but also there are muscular degenerative diseases as well, are a group of diseases. There are more than 30 of these known now. They're called nucleotide repeat expansion disorders. Again, genetic, either involving neurons or muscles. These are some well-known examples of these diseases. And they all involve a simple nucleotide repeat that gets copied many, many times within a gene. And as I said, the repeats are simple. They're often triplet repeats. They tend to be GC rich. And just as an example, in the Huntington disorder, there's the Huntington gene where there's a CAG repeat that in some families becomes amplified many times. And a characteristic feature of all these disorders, which I'll come to as an important point in this talk, is that you have to reach a certain number of these repeats before the disease becomes manifest. So in the case of Huntington, below 20 is generally asymptomatic. And if you have a repeat copy of this triplet beyond that, then the disease becomes evident, and with increasing copy number, more severe. All of these diseases are currently untreatable, which is also, you know, you'll see where this story is going, but I think, you know, maybe we have at least a new hypothesis that could be at least tested to see if it may be applicable even down the road to potential treatment of these diseases. So this just shows some more of these repeat expansion disorders. making the important point here that these repeats sometimes occur in an exon, so in a protein coding region. That's the case of the Huntington disease. But in many other cases, the repeat expansion occurs in a non-coding region of the RNA. In fact, that's even more common. They can occur as in muscular dystrophy at the 3' UTR. They can occur in introns, as in the case of ALS. They can also occur in the 5' uncoding region. And all of them, again, have this characteristic phenomena that you need to reach a certain threshold number of these repeats before disease becomes manifest. So let me just tell you kind of where the field is right now. Of course, this is a big field, lots of research on it. There are three general thoughts on how disease could originate. And I do want to stress none of these need be mutually exclusive. And in fact, quite likely, like many things in biology, it's probably not either or, but an and phenomena. But so one, in the case where the repeat occurs in an exon, as in case of Huntington, in that case, the ribosome copies that at a start codon and produces a polypeptide that has a stretch of polyglutamines. And the polyglutamines are a aggregation-prone sequence, and that's thought to give rise to an aggregate of the Huntington protein. But as I mentioned, many cases the repeat is not in a coding region, but there's an alternative thought that there's a non-canonical translation phenomena, which is called RAN translation. And the mechanism of this is not very well understood, but it's thought to involve ribosomes translating without a start codon, which could include an intron or other non-coding region. And running through this repeat, it would give rise to a repeating peptide. And it's thought that these peptides might be toxic. So these are called RAN peptides. There's another school of thought that the RNA itself may be toxic. And one thought is that these repeats create, through Watson-Crick base pairing, hairpins. And these hairpins are attractors for various RNA binding proteins that get sequestered on these hairpins. And also that once the proteins get onto the RNA, and it's usually been thought to be more of a protein-based phenomena, that nuclear aggregates can form. The RNA can aggregate with the protein inside of the nucleus. And I guess I'm supposed to point with the little pointer, but this is taking too much work. And I can't even find the pointer. So maybe... it was visible. Yeah, the home audience will just have to imagine where we're pointing. But anyway, so this is an in situ hybridization of the RNA, and you can see that there are foci present. In fact, at biopsy, this is a very prevalent phenomena of the pathology of essentially all of these repeat expansion disorders. And this just shows in situ RNA hybridization for a variety of different diseases where nuclear foci of the RNA have been reported. So the real questions that I want to talk about here focus on these foci. And first of all, the issue is what causes their formation in the first place. And second, the foci also form when the repeat number reaches this critical threshold as well, similar to the disease. So why are these foci only seen after a critical threshold? And maybe the biggest question of all is why did we even start working on this in the first place? As you heard, we're working on lots of other things, microtubule motors, but these this neurodegeneration phenomena was, was previously not a subject in the laboratory. But I think it's a good example of really just how research works and how you can get tossed into completely new problems and new fields. But I guess what got us interested in this originally really was from immunology and T-cell signaling, which our lab does work on. But there are foci of a different sort that are present in the T-cell signaling pathway. And not that you need to know all of this. This is just a lot of players here. But when people look at many of these proteins in the T-cell signaling pathway by GFP or immunofluorescence, but what you see, like for the T-cell receptor, are after signaling begins, you see the TCR form little puncta in the plasma membrane, or foci, if you like. And this is true, in fact, for all of these components in the T-cell signaling pathway. And there was one protein of particular interest to us, which is this LAT protein, which classifies as an adapter protein. And with Mike Rosen and other colleagues, we began to do some of the biochemistry of this LAT protein in vitro. And what you see is a really remarkable phenomena where effectively foci of this LAT protein form in a completely purified system where we have LAT not on the T-cell membrane but on an artificial lipid bilayer in a phosphorylated state. And we now add a couple adapter proteins that bind to LAT. I'll explain them a little more later. But after you add these proteins, what happens is the LAT forms these effectively phase-separated particles, and it's completely reversible. So in this case, this is not an aggregation phenomena, because if you add phosphatase to remove the phosphates on LAT that are the binding sites for these adapter proteins, these phase-separated objects kind of disperse and become monodisperse again. So what's happening here is you have a multivalent protein, which is LAT. It has many phosphorylation sites, tyrosine phosphorylation sites, that act as binding sites for a bivalent adapter protein that binds to the phosphate. So you can, and that binds to another multivalent binding partner, which is SOS. So you can see here how you can build up kind of larger networks of essentially a larger protein network from these components, from these multivalent interactions. And Mike Rosen's lab really described this phenomena very beautifully from his lab, where he even developed ways of studying this with kind of artificial polypeptides, where you have for example, a polypeptide that has multiple SH3 binding domains interacting with another polypeptide that has multiple effective binding sites, which is polyproline motifs. You mix these things in a test tube, and they phase separate into droplets. And they are liquid-like. In fact, these droplets, which are just made of protein, actually fuse with one another. So they behave very much like oil and vinegar on a dish. And an important part of this phenomena is that it's very sensitive to the valency of the interaction. This is important because I'm leading up to this for the RNA as well. But these show phase diagrams. So this is just a way of showing whether the material is homogeneous or has become separated into two phases. So you increase the concentration of one protein versus the other. And then the blue dots are homogeneous. There's no phase separation. The red dot is when you now see these two states, and that you see a phase-separated droplets begin to form. And you can see, first of all, it's very abrupt. You can go from small changes on this concentration curve, and it's essentially an all-or-nothing reaction. The second thing is it's highly dependent upon valency. You can see here there's no phase separation. When you increase the valency by 1, you now get into a phase-separated domain. 1 more, and now essentially everything phase separates, even at the lowest concentration. So, well, it's a long story. I'd be happy to talk over at Paris a glass of wine of how we actually really got into this. But we did begin to think whether, or particularly Anker, whether that phenomena that I showed you was attracted a lot of attention from the protein community. But the question was, would this be applicable also to nucleic acids as well? Now, traditionally, if you look up reviews in this field, as I showed you, this is the way people were thinking about foci formation, that it was really a protein-based phenomena that leads to these foci. But what Anker began to think about and realize is this is the Watson-Crick base pairing that was usually described, that it would form a hairpin. But if you have many of these repeats now on a single nucleic acid, it seemed possible to Anker that you would not only get intramolecular hairpins forming, but you could get intermolecular interactions between different RNAs that are interacting in effectively a multivalent phenomena where you have multiple binding sites between multiple RNAs that begin to interact with one another. So it was really a very, very simple idea. In fact, it's somewhat remarkable that really no one was thinking about this at all, that everyone was drawing this as a intramolecular interaction. So Anker then, first of all, began to test this idea, whether this may be happening and may be relevant, and tested whether purified RNA could undergo a phase separation phenomena, similar to what I showed you for the proteins. And so we did this with in vitro transcribed RNA, pure RNA, no protein. And indeed, if he looked at a triplet repeated 66 times, fluorescently labeled RNA, it indeed phase separated. This shows another repeat for muscular dystrophy. But as a control, if he took an RNA of the exact same length, the same GC content, but randomized the nucleic acid sequence, instead of having this repeating structure, he found that under these conditions, the RNA didn't phase separate. So it was a sequence-specific, repeat-dependent phase separation phenomena. Now, before I also showed you this perplexing result in the field, really, that you needed this critical threshold to get foci and to get disease. And as I described to you, it's well known generally in phase separation that there is this very strong dependency on valency. So Anker tested this naturally, and again, found a very striking dependency on the number of these repeats to get this phase separation phenomena. So when there were 20, there was no phase separation. And again, abrupt transition to where these RNAs started to phase separate. Although this admittedly very correlative, but the repeat length where these phase separation occurred is not dissimilar to what's noted for the threshold where the Huntington disease also begins to occur. So another very well-known nucleotide repeat, many of them are triplet repeats, but there's one for ALS, which is a hexanucleotide repeat of 4 Gs and 2 Cs. This is the most common familial form in ALS and frontal temporal dementia. It's not the only genetic cause. There are other causes as well, but this is the most prevalent. Again, a threshold requirement. But in this case, and this was known in the field before, there's another kind of nucleic acid interaction called a G quadruplex. And this is basically a planar arrangement of 4 guanines that are hydrogen bonded in a planar network. So not the typical Watson-Crick base pairing. But again, the way people usually describe these quadruplexes as forming is where all the Gs are contributed by a single RNA. So effectively, again, an intramolecular G quadruplex. But, you know, Anker also reasoned that why do all the Gs have to come from one RNA? You could imagine having Gs coming into this quadruplex from other RNAs as well. So again, he tested this RNA, and again, it formed this repeat number dependent phase separation phenomena. Now, one thing I'll also add about the RNA in vitro is, unlike the protein that I showed you, most of these RNA particles that phase separate in vitro are solid, or at least become solid. As I'll tell you, I think they are initially liquids and then rapidly, due to solidification of base pairing, become solid-like. And the fact that they become solid-like is shown by this photobleaching experiment, where you photobleach part of these particles, and you can see that this bleach does not recover. If the RNA molecules were mobile in those objects, then the fluorescent RNA would invade the dark area, and you would see that recover. But that doesn't happen. But we think they were initially liquid-like because you often see, first of all, a spherical shape is really indicative of a liquid-like behavior. But also, you would see some droplets that were in the process of fusing, but then appear to be solidified before they can relax to a spherical geometry. Okay, so that's the work in vitro. Of course, the main question is, does this have any relevance out of a test tube and in a cellular environment? So Anker then tested this with the following kind of a construct and assay. So he expressed an artificial transcript here, which he can control with an inducible promoter. It then had stop codons just to prevent any possibility of normal translation. He then had a test sequence, so you can put in any repeat you want or any number of repeats. And then at the very end, there was a visualization element, which is essentially a series of hairpins from a viral coat protein called MS2, which is a target for a protein which we could fluorescently tag, which is the MS2 binding protein. This was originally developed by Rob Singer. It's basically a way so you can see your RNA in a live cell imaging experiment. And this is what it looked like here. If you induce transcription with a small number of repeats, no foci, all the RNA goes to cytoplasm. If he now adds the large number of Huntington repeats, he now sees these foci form in the nucleus. And this is what it actually looks like in a time-lapse movie after gene induction. You know, you can see these foci, these RNA begin to aggregate within the nucleus. Now, interesting, unlike what I showed you in vitro, in cells, these RNAs are liquid-like, or more liquid-like anyway, because if you photobleach them, you can see them recovering over here. They also behave liquid-like because you can see them fusing. Like you can see a couple RNAs here that are kind of gobbling themselves up to form larger particles. Now, what we think is happening is that there are cellular factors, unlike the in vitro situation where there's no protein. here we think the RNA is phase separating, aggregating, but it's not alone in this environment, and there are proteins that are in there working on these RNAs, probably trying to solubilize them. We know that there are a huge number of protein chaperones that are constantly folding proteins, and there's probably a whole world of RNA chaperones that we still very poorly understand, but some of them are likely to be helicases, or at least we suspect that they are, because if we do this photobleaching phenomena like I showed you here, normally they recover, meaning that they're liquid-like, but if we take away ATP from the cell and now we photobleach them, there's no recovery. So they're behaving like solids again. So there are probably some interesting ATPases that are working on it. So of course the real question is, can you disrupt these aggregates? And one strategy is, if there's a multivalent phenomena that are bringing these long transcripts together in an aggregate, one way you could potentially compete for that is by adding an antisense oligo, because those are short things that are going to be binding to the repeats, and they are effectively competing for these intramolecular interactions between these long transcripts. And if you do that, add antisense oligos, the repeats don't form, or they will dissolve. The other thing that we found that will dissolve them are molecules, small molecules that intercalate into duplex nucleic acid and effectively block RNA base pairing, and or DNA base pairing in this case. One of them is a cancer drug called doxorubicin. It's widely used in cancer. It will not be a great drug for neurodegeneration, so it's not being used here for that. It's way too toxic for multi-year treatment. But the point is that doxorubicin dissolves these RNA both in vitro and also in living cells. It at least illustrates the possibility that one can potentially develop small molecules that could affect this phenomena. And also doxorubicin works on foci from patient cells as well. So the bottom line summary is that we think that RNA can undergo a phase separation or / aggregation phenomena that is also built upon is sequence specific and occurs when you get these repetitive RNA sequences, and that results in aggregation forming in the nucleus. I think where the work is leading to are several, I would say, big questions. So one is, I talked about a sequence-specific RNA that's associated with disease and causing a pathological foci. But we know that cells actually have a lot of other RNA protein non-membranous granules. Some are called stress granules, some are called P-bodies. There are things in the nucleus called speckles and other paraspeckles and other RNA protein bodies. Most of the focus in these fields has been on phase separation of the proteins. But this does raise the question whether there are some RNAs that are meant to be phase separation prone, possibly working with proteins to lead to at least some of the phenomena of normal physiological ribonucleic acid protein particles that form in cells. The other big question is, I've told you nothing about the disease, and indeed we don't have any evidence for whether the phenomena of foci formation is connected to the disease. This is a big open question. I think this hypothesis that I presented to you at least offers, and the possibility of also making drugs that could affect these foci, at least offers new ways to test whether these foci might actually be contributing to disease formation. So anyway, with that, I'd like to thank Ankur. And I still have some time. Are people patient for like 20 more minutes of a completely different topic? Okay, great. Well, so we're really going to have to switch mental gear here because we're going from cell biology, from, real mechanistic biochemistry of phase separation to thinking about organ and tissue morphogenesis. So this is a project done by, again, really fantastic and daring postdoc. I say daring because she came from a biochemistry lab working on kineticore biochemistry, really hardcore biochemistry, and then switched to working on organoids and adapted to this in record speed with a lot of help from a great colleague at UCSF, Ofer Klein, who helped with all of the mouse work and a lot of advice in this project. Now, the work I'll tell you about involves the intestine. And one of the interesting parts about the intestine is that, first of all, there are many different cell types in the intestine. Second of all, it's an organ a tissue that is constantly turning over. I think there are about, on a daily basis, I've read there are around 300 grams of intestine. Oh, you probably know, Danielle knows this topic. You should give this part of the talk here, Danielle. But large number of these cells are turning over on a daily basis. And so most of the active cell division is occurring at the crypts here, where the stem cells are located. Eventually these cells migrate up to the villus and they're sloughed off at the very tip, and there's this constant process of cell formation and renewal going on. Now the topic that I'd like to take up with you today is this very interesting patterning of different cell types actually throughout the intestine, but I'll particularly focus in the region of active cell proliferation. And this just shows an image of two really critical cells. One is the stem cell population, which are marked by a certain receptor called LGR5. And the other group of cells are shown in red, which are Paneth cells, which are a secretory cell line. And these Paneth cells are thought to create a niche, an important niche, contributing Wnt and other factors for the stem cells to allow for their continual division. And what you see here is that there's this intermixed pattern of the Paneth cells and the stem cells in probably a way that, that may be actually very important for creating this niche for continual stem cell division. But the question is, how do you get this alternating pattern, and how do you maintain it? Because it's a non-trivial question to think how you maintain this pattern in a zone where there's continuous cell proliferation. Because just based upon the notion of cell proliferation, what you would expect is when cells divide, they create patches. They don't create this alternating pattern that I just showed you. And in fact, in many kinds of epithelia, you do find these kind of patchy networks of cells from clonal analysis. This is just from a clonal analysis in a Drosophila follicular epithelia, where, again, you see patches. You don't see this alternating pattern. So the question is, how are this alternating cell type pattern generated in the intestine? And the bottom line, this is the answer, and then I'll back it up, is that cell division itself is creating a cell dispersal phenomena that is giving rise to this pattern of different cell types. And I'll explain how cell division leads to this cell dispersal phenomena and this alternating pattern as I go forward. So what Kara did in this case is to try to understand this question of this patterning is to look at intestinal organoids from adult mouse. And this is really following a lot of beautiful work that was pioneered by Hans Cleaver and his colleagues. And I guess what we brought to the equation is good imaging, which is something that our lab does have to offer. And so we can image these organoids for, even 1, 2, 3 days in culture. And what you see here in that movie, maybe I should just play that again, is a 12-hour movie. Whoops. And all of these little spherical blobs that you see here, I'll show you, are cell divisions that are going on. So this organoid is very alive, undergoing lots of cell divisions. And we'll explore how those cell divisions work. So one thing, first of all, is the intestine are columnar epithelia. The nuclei normally in a non-dividing cell are located towards the basal surface. The interior surface is the apical surface here. And it's been seen for decades, so this is nothing new, but even in decade-old histological sections, you can see that if you stain for nuclei, the cell divisions, the chromosomes, are dividing at the apical surface. And you can see this in movies that Kara has taken very nicely of histone GFP. The nuclei come down, they divide at the apical surface, and then they go back up. So now let's look at a movie of where we can see these two alternating cell types here. The Paneth cells, the secretory cells in red, and in green is are the stem cells. And we'll follow one division where you can see now that cell comes down to the apical surface to divide. And now the two daughters come up. And as the two daughters come up, they're separating, and they're leaving a stem cell in the middle. So they've created an alternating pattern. Kara's done a lot of statistics on this. I'm showing you one example, but indeed this only happens 50% of the time, pretty close to 50%. However, if you now follow that, if you follow daughters that didn't separate on their first division, and you follow them over time, the neighboring cells, when they divide, they have a 50% chance of inserting between the non-separated daughters. So if you follow these cells over a few divisions, these cell divisions will tend to separate the daughters in this kind of pattern that you see. So what I'd like to do now is really go through the mechanism of what's happening. I kind of gave you a superficial overview, but the first question is, what is the mechanism that causes this apical mitosis here? And then the second question is, how is it that these daughters are separating during cell division? So nuclear migration to the apical surface is nothing new. There's a large field on this. Probably the most studied system is in the neuroepithelia, which has been examined quite a lot, where this gives rise to cell divisions in the brain or elsewhere in the nervous system. But basically, in this case, the nuclei kind of go through this pattern of movements between S phase and coming down to the apical surface, closer to the apical surface at G2, and then at the very apical surface during mitosis. Most of this process over here for the neuroepithelia is driven by microtubules and microtubule motors. This whole phenomenon, by the way, is called interkinetic nuclear migration. That's the word used in the field. What I'm going to tell you about, and I'll show you the evidence, and it's thought that the last part of this process here is driven by actin. What I'll tell you in the epithelia, microtubules are not involved at all. And likely this last actin step, which may also be applicable in the neuroepithelia, is what's occurring in the intestine. So what cells do when they undergo mitosis is they round. This occurs in all cells in tissue culture, basically fairly universal phenomena with mammalian cells. And intestinal cells do the same thing. You saw that in the movie. The cell rounds before it comes to mitosis. In the case of your cell in tissue culture, it rounds it rounds onto the glass surface. That is pretty obvious because the cell is adhered to the glass surface. In the case of the intestine, the cell rounds onto the apical surface largely because it's connected to its neighbors. And it's connected by adherens junctions, which are basically collecting all of the epithelial cells together to create a barrier. This is how the intestine creates a barrier through these adherens junctions at the apical surface. These adherens junctions are maintained from interphase to mitosis. This has also been known before. So when the cell rounds, it basically has nowhere else to go but to the apical surface. This is where it's locked down, where the mitotic cell is locked down between its neighbors. And I'll also provide some evidence for that, largely based upon some pharmacology studies, where we've looked at different drugs to see which cytoskeletal systems are involved in that apical mitosis. And so if you normally do this in a control cell, you can see the DNA divides at the apical surface. If you add latrunculin, basically the cell actually is dividing. Now the chromosomes are dividing not at the apical surface, but at the basal surface. The cell still retains its columnar shape. If you knock out all the microtubules, the cell doesn't care. It still rounds, and the nuclei comes down to the basal surface. And this is just quantitated here, where one's looking in mitosis, where the nucleus is compared to where it started at the basal surface versus the apical surface down here. And you can see that in control microtubule case, exactly the same. So the phenomena that's driving the apical mitosis is basically cell rounding onto the apical surface due to the adherens junctions. And that process of apical rounding also is critical for establishing the plane of division, where the spindle is oriented parallel to the epithelial plane, and that's necessary to create two daughters that are oriented along the plane of the epithelia. If you have actin inhibitors and you prevent that apical rounding, the chromosome and the mitotic spindle is oriented in the completely wrong direction, 180 degrees in the wrong spot, and that's because the spindle is still adopting the columnar shape of the cell. So this is the really, perhaps even the more interesting question. How is it that these daughters separate from one another? And two key aspects of this. So first of all, this is a movie with a myosin light chain. So this myosin here is the contractile element that's driving the last step of cytokinesis. So it's a good way of visualizing where the contractile force is coming from. And if you watch this, the furrow ingresses in one direction, basically from the basal surface to the apical surface. It's completely asymmetric. Normally in a cell dividing in culture, the furrowing of cytokinesis occurs basically symmetrically around the chromosomes. Here, it's ingressing only from the basal surface. Again, the reason why we think that's true is at the apical surface, all of the actin and myosin is tied up in a very large circumferential ring that, again, is maintaining the epithelial barrier and the connection with its neighbors. And that's what gives rise to this asymmetric cytokinesis. And this is the key point. At the point of this asymmetric cytokinesis is where a neighboring cell basically has a wedge to insert between the two daughters. And this is what you can see here in this movie. We've now, Kara's marked the two cell types over here. So in this case, the stem cells are the green cell, and the neighboring cells are the secretory lineage over here. And just to show that again, once that furrow opens up, nature abhors a vacuum, and the neighboring cell basically finds an opening and inserts itself in between the two daughters. And the last thing to say about this, the last step then, is how the system resets itself, and how the two daughters then get back to the original shape. And that appears to be an actin-driven phenomena very similar to cell migration. This is a recent daughter here that's going to migrate back to the basal surface, and it extends a lamellipodia and basically is crawling up against its neighboring cell, but very much like, you know, a crawling migratory cell, and that's how it reinserts at the top. Again, the pharmacology suggests that this is all actin-driven. It has nothing to do with microtubules. And to show this, Kara did a very nice experiment where she arrested the mitotic cells in this organoid with inhibitor of the spindle assembly checkpoint. So it basically holds the cell in mitosis. She can wash out the inhibitor in the presence of drugs or in a control situation, and then ask, after the cell exits mitosis, how does it come back up to the basal surface with these different drug treatments? In the control case, comes up very nicely, as you can see. In the latrunculin case, the nuclei never come up. They're stuck, basically, at the apical surface. And in the case of the microtubules, they basically come to the very top. It's unaffected. You know, this is basically the cycle that I just showed you. We think that the actin-driven rounding is what causes apical mitosis. This asymmetric cytokinesis is what allows daughter cell insertion to drive the separation of the two daughters. And then there's actin-driven protrusion that gets the daughters back up to the cell surface. And the last thing I'll just say in a couple slides is that in this model, the position, the cell shape here is columnar, and basically the nuclei are adopting two different positions, a basal position or an apical position. And that shape, one might expect from that last model to be important for daughter cell dispersion. You need to create different layers to get insertion from neighbors to cause dispersal. So Kara tested this by creating organoids of different shapes. So one way she could do this is to make organoids from fetal intestine as opposed to adult. Those organoids basically form spheres with very different shapes. Instead of being these long columnar cells, they're exactly the opposite. These are just a Z section here showing the top of a spheroid and the side. And instead of long and narrow, they are short and fat. And when you look at the cell divisions in this case, you see that these cells divide. And this is very typical. no daughters insert in between them. Effectively, there's no daughter cell separation in these fetal spheroids. And another way that Kara looked at this is, even more interesting, I think. She, under the same conditions, could create organoids of different shapes. And that was done by adding adult organoids with a high concentration of Wnt. At very high concentration of Wnt, you get two different kinds of organoids forming in about 50-50 frequency, either spheroids or these normal-looking intestinal organoids. And if you look at how daughter cells separate in this case, and this shows actually after a few cell divisions, effectively a clonal kind of analysis. In the normal columnar organoids, you can see that the secretory cells and the stem cells have this alternating pattern. But in the spherical case, exact same conditions, just different geometry, you get effective patches of these cells. The daughters aren't dispersing. And just to say, I don't want to go into in great detail, but we're also doing mathematical simulations to actually play this out for doing mitoses effectively in silico with different cell shapes. And the results actually come out very, very similar in this in silico model, comparing daughter cell separation and cell dispersion as, and here where they're not separating and you get clonal patches, very similar to what we see in the native organoids. The last question I don't have an answer to right now, but maybe will in a few weeks. The other question is this a more general phenomenon? We love the intestine, don't we, Danielle? The intestine is great, but some of the principles that I just outlined may not be specific just to the intestine. There are other cases where there are other epithelia where cell dispersion may be very important. This is actually a very old classic experiment of clonal analysis in the epiblast, so very, very early embryo. And when a patch of cells were basically transplanted in that embryo, after many divisions, you can see that those cells disperse. And this is also in a columnar epithelia. So we now like to look at this to see how are these daughter cells separating, and are they separating by the same mechanism that looks like what I described in the intestine. So last slide. Thank you very much for all of your attention, by the way. I know this was a little long. But again, just absolutely fantastic postdoc. Our lab, had never seen a mouse before, never worked with organoids. And so all this work was really done by Kara with a lot of help from Nico. I forgot to say a lot of the imaging here was with a home-built light sheet microscope. And Nico was a developer of that microscope in the lab. And I've already mentioned Ofer and Ofer Klein. And thank you so much for your attention. It's great to be back in Paris and at the Curie. Well, I saw your hand first, so I'll take.
I have a question about the invasion. So when the neighboring cell invades between the dividing cells,
Yeah.
Does it detach from the apical? Does it detach its apical surface, or does it kind of come up from the basolateral side and then re-specify where the apical side is?
Yeah, it's a good question. What happened? We're analyzing some of those movies now to better understand that. The apical surface is so small that getting really good 3D movies of that to look what happens. It ultimately has to pry apart the apical surface as well. What we think right now is it is a basal invasion that gets propagated to the apical surface. And the reason why we think that how this basal insertion also happens, why it happens at the basal surface first, is the basal surface is very weak, cortically weak. The evidence for that is I don't have a movie of that, didn't show you the good movie of that, but many of the cells that are undergoing cytokinesis, the basal surface is blebbing. And blebbing are these little, like almost defects in the membrane that pop out. And a lot of other work has shown that blebbing occurs when you have a cortically weak plasma membrane. So it's almost an indicator of a mechanically weak surface. So that blebbing phenomena is seen, you know, at that basal region. The apical region is like a rock, as far as we can tell. There's no blebbing there. There's a persistent actomyosin band that's around the entire perimeter. And actually, another point, maybe Danielle has comments on this too, or we can talk later. If Kara's also measured the apical perimeter of the dividing cell in comparison to the neighbors, and the apical footprint is essentially identical to the neighbors. This also at least suggests that the apical, the kind of the apical pulling forces, if you like, are equally strong in mitosis versus interphase, because otherwise the interphase cells would be tugging away at the mitotic cell and changing the footprint or pulling that cell apart. So it's almost like that apical footprint is a good measure of how the you know, how all the cells are tugging at one another. And it looks like the mitotic cell at the apical surface is identical to its interphase neighbors. So that surface seems to be really mechanically rigid and unchanging during the whole cell cycle. So it's only this basal surface. But the point you raise of eventually how do you get something into that apical surface is a great question. So don't have the full answer on that yet.
If I may intervene in that discussion.
Yeah.
There is another phenomenon different from what you describe.
Yeah.
Which showed, actually, I would say that the apical surface is highly dynamic as well.
Yeah.
If you get an infection of the gut by salmonella or some other, there is neutrophils.
Yes. Mm-hmm.
That actually crawl between the cells, open. Let's say not only you have adherence junction, but you have tight junction, which are even
tighter.
You know, tighter. And the neutrophil managed to squeeze in between and finally crawl on top of your apical surface. And people have measured the electrical resistance during that process, and there is no leak. Fortunately, there is no leak.
Mm-hmm.
But yet there is a whole cell that managed to get through
mm-hmm.
by something which is very dynamic. So I think this is probably, there is something equivalent.
That's a good point.
I will believe that they go out
Yeah.
and maintain some contact in between with, and then they finally reinsert. They never get, they are never free, otherwise they will be lost.
Right, right.
I will have question later.
Yeah. Yeah. No, so that's a good point. I mean, maybe this basal cell insertion is analogous to a neutrophil getting, you know, in between neighbors. It's a great point. Maybe here, since you're, and then, yeah.
Well, yeah, I had an RNA question.
Yeah.
So actually 2 questions. So one is about specificity and the other is about quantity. So first, how much you think the actual sequence matters for formation of these aggregates as compared to as long as you can make a loop and you have enough repeats, you will make an aggregate. Like for instance, do you think your MS2 stem loop, provided you put enough of them, they would make a similar type of aggregate that would behave similarly? And the other question is also how much of the stuff you actually need in the cell to be expressed, and whether this is consistent with the expression you see in disease, for instance.
Yeah, well, good questions. I mean, Sequence is important because obviously we do controls with the MS2s and some other random sequence upstream of the MS2, and there's no aggregation. So that phenomena is dependent upon you putting that triplet repeat in front in that transcript. I mean, that's one additional thing that we are trying to study right now is, in addition to the repeat, whether there are any cis-acting sequences in native transcripts that are modulating whether that repeat zone is going to be aggregation prone or not. So there may be a bigger story than just the repeats, but for sure not everything aggregates. The second question was, I forgot.
How the expression you would need in a cell to make this relate to the disease?
No, that's a good question. Some of these transcripts are very, very low. We've titrated down our viral titer, again, fairly low, and we still see it at the lowest viral titers that we have. Whether we've titered it down, you know, to identical levels in some of these transcripts, probably an open question and a fair question. In general, what we find for the phase separation is it's highly dependent on repeat length, but not so much on concentration. So once you're in the zone where there are many repeats, we've decreased the RNA concentration in vitro very, very low. So most of these objects that phase separate are extremely low RNA concentrations. They're not obviously down to single molecule because you need multiple copies, but they're forming diffraction-limited spots. So we have titrated it down in vitro quite a long way, and you know, we still see this phenomena. By the way, if anyone wants to go, maybe I'll take 3 more questions and then stop, and I'll be short. So maybe 1, 2, and 3. Okay? Yeah.
So I would like to ask a question related to the second part. So what would happen if you actually change the orientation of the cleavage plane, for example, by using the latrunculin A or mutations that misorient the mitotic spindle? Would you lose the separation between the 2 cell types? And second, what is the explanation for only having 50% of the divisions resulting in an inserting cell?
Yes. Okay, both great questions. First question, I don't know. It's not trivial to wash out latrunculin. So as far as I know, I'm not...
Basically, the question is, could that be like a repulsive mechanism between the 2 equally created daughter cells, allowing another cell to penetrate?
I mean, when there's a... if the 2 daughters separate and form 2 cells in the... Yeah, I have no idea. It's a great question. I'm not sure what would happen, or whether also the daughter may just refine. You know, there's one daughter that's going to be, it's going to be a situation where other stem cells, there's one attached to some niche region and the other one divides and migrates off. So I don't know, in this case, maybe the daughter will actually, I don't know what will happen to the daughter. Maybe it will refine the apical surface again. I don't know. It's not trivial to get latrunculin out. I mean, but I don't know that answer, and I think it's a worthy experiment. So we should at least attempt to wash out the latrunculin. And the second question is, why is it 50%? I think it's I think the thing I want to stress is it's not exactly a completely purposeful event. It's like a weak. I think the answer is there's some stochasticity to it, whether that the neighbor is actually going to pry and wedge itself in between the 2 daughters. It's not like some precise signaling pathway where series of events are happening leading to a very uniform outcome, it does appear to be a stochastic event, whether that separation is going to occur at that division. As I said, if you follow these cells over multiple divisions, each event cumulatively tends to disperse these cells. I think to some, the other answer is like a teleological one, is why do it in the first place? Maybe even a better question. I think to some extent the phenomena of having this apical mitosis, for all the mechanical reasons, and maintaining a constant barrier function of the epithelia, which you don't want to lose in mitosis, right? That would be a bad thing. So just maintaining that constant integrity of the apical surface throughout cell divisions alone probably just mechanically leads to a dispersion phenomena happening. I mean, that's part of my thinking of this. Now, cell biology and development also may take advantage of that mechanical event to build, in this case, in the intestine, kind of niche structures around this phenomena that is happening, which is, not necessarily built for, I would say, stem cell function or differentiation, but because this is happening in a dividing tissue, it may be that the system has also evolved to create these niche relationships between these secretory cells and these stem cells, because it's very compatible with this kind of mitotic-driven dispersion function. But, I think it's a reasonable point. But the point is, it is stochastic. It's not a purposeful event that's happening here.
I have a question related to the first part, the RNA aggregates. So what do you know about the stability of your artificial RNA aggregate when they are formed in the cell? And did you compare their behavior or their fate by doing a comparison between the cells that can divide versus cells that cannot divide?
Yeah, we haven't really followed very long term the lifetime of, what the half-life of the RNA is. In general, well, we're testing more constructs right now in different lengths and so forth. The ones that I just showed you here and the ones that are published, the cells actually do live with these RNA aggregates for many days, like a few weeks. So it is like a stable, relatively, after induction, it's a fairly stable structure. It's not doesn't seem to be turned over very regularly, and the cells seem to be dealing with these nuclear aggregates as well. Maybe last question, and I'm happy to answer any more questions later, but maybe this will be the last one for the poor audience here.
I have a question about the second part.
Yes.
So if you think that the cell is rounding up, and so it comes from the basal surface towards the apical surface, and it's a passive process, how does the reverse process takes place? Like, how does the polarity is induced so that it goes in the other direction? The other question which I had was, if somehow one is able to change the aspect ratio of the cells, so by confining or in some other way, do you think what would happen to the division?
The first one, really don't know the answer to that. It would be very interesting to look at polarity proteins in that, or even phosphoinositides. I mean, there is a large literature of what the leading edge of a migratory cell looks like molecularly. There are a number of polarity proteins and proteins that define a leading edge actin cytoskeleton. All we could say is it looks very much like the behavior of a typical leading edge that one sees in other migratory cell types, but we haven't looked at it at molecular composition yet. And the second question was, oh, I'm sorry.
About
what?
the change in the aspect ratio. And
Yeah. Sorry. Well, I kind of rushed through that because it was getting late. But this is a change in the aspect ratio. So this is the exact experiment that you're describing. These cells are, so this is a spheroid. So imagine this being a sphere. And you're now looking at it from one z-axis and 90 degrees. So this is cutting a slice through that sphere. So you can see that these cells, instead of being very long and narrow, are short and wide. So it's exact change in that aspect ratio. And you get a completely different outcome. You get, not surprisingly, the cells, the daughters are still dividing like that. They're dividing in the plane of the epithelia. In fact, that's very permissive for that because it's already oriented in the right direction. But there's no cell invasion happening in this system. So that aspect ratio is very critical. And one of the things that we're doing computationally in silico is to change that aspect ratio, and at least in a model. I mean, here we have two states, but we can't fine-tune the aspect ratio beyond what you see here. But in silico, we could start asking at least some of those questions and see like what aspect ratio is really consistent with this cell dispersal driven mitosis. Normally it's about a 2-to-1 ratio for these cells, but so the question is, how forgiving is that? If you change that a little bit, how does that change the result? So anyway, testing that in silico. So I think you have a question? Okay, so always one last question for the director here.
Okay, in fact, I have two.
Yeah.
One which has to do with the ATP that you were saying in the first part of your talk with respect to the RNA and potential role of helicases. I was just wondering whether instead of helicases, what is actually needed is a phosphorylation state. And so have you considered that from that angle?
You're correct. It could be interpreted from that angle. So no, we don't have any evidence for that. I mean, right now, one thing we're doing is we are trying to test various helicases and see if they, are modulating the formation of these foci. But you're correct. We don't, from that experiment, have a direct link that, that the ATP effect is due to a helicase. It is due to helicases and not a kinase.
Really interesting, perhaps, to think about it from that perspective.
Absolutely.
Perhaps by playing in a semi-in vitro system using hyperases and gamma ATP, so that you could have phosphorylation that is stable, but then see if that does something or not. I mean, there are ways to look into it. The second is perhaps a stupid question. One thing I don't understand with the aspect with the mitosis that you were describing is how the cell that goes down and divides can then have a way to generate an alternating pattern if you don't have concomitantly division on the one that are on the top, because the ratio is not right. At some point there may be a problem. So how is there a synchrony between this? How do you generate the pattern? Because that's not enough to go down and divide and then get back. You also need to have...
No, I mean,
you see my point? Maybe it's stupid.
I think I see your point. I mean, there are two elements of the question. One is, like, what is your relationship between your neighbors?
Yes.
And then the other question is, like, who who exactly are your neighbors? I mean, what is their identity? I mean, obviously, you can have daughters separated by neighbors of the same cell type, right? And that will disperse the daughters by a mechanism, exactly. But it won't give you the pattern. So the pattern does require also creating the right ratio and maintenance of stem cells and Paneth cells and all of these things. So that is a secondary layer to the problem. That's a secondary layer which is really a cell differentiation question.
Exactly.
Hans Cleavers probably can tell you the answer to that one. Yeah, that's an even more complicated question because, technically what we see also in this experiment are secretory cells. It's a secretory cell lineage, which are, in fact, the marker we used is indicative of many secretory cells. So we can't distinguish between a secretory cell precursor, a Paneth cell, or something else. But also in the adult tissue, the Paneth cells that are at the base are actually thought to be derived much higher up in the epithelia, and then through a mysterious process that I have not seen described in the literature, are thought to migrate down. Well, most of the other cells are actually migrating up. That cell is actually migrating down. So in reality, you know, that ratio of different cell types there is, I do think, involves cell dispersion, so you don't get clones, but it also does involve generating the right differentiating pattern when stem cells divide to create new progenitors, and it also involves cell migration. So getting the absolute right pattern and ratio and numbers of these cells at the base, as far as I know, is still a fairly big mystery. So we just tackled one part of that mystery, but that's like not a dumb question. That's like the big picture question, actually. So anyway, thank you very much for your time.